By 1953, scientists already knew that DNA was a double helix made of two complementary strands, but one huge question remained. How does DNA make an exact copy of itself before a cell divides? Nobody knew the answer. Scientists proposed three different models. The first was conservative replication. The original DNA molecule stays completely unchanged, while an entirely new DNA molecule is made. The second was semiconservative replication. The two DNA strands separate, and each old strand acts as a template to build one new strand. The third was dispersive replication. The DNA breaks into pieces, copies are made, and old and new DNA become mixed throughout both strands. All three seemed possible, so how could scientists determine which one was actually correct? Matthew Meselson and Meselson Stahl designed a brilliant experiment using E. coli bacteria. First, they grew the bacteria in a medium containing heavy nitrogen, called nitrogen-15. Since nitrogen is a component of DNA bases, every newly made DNA molecule became heavy. Next, they transferred these bacteria into a medium containing normal nitrogen-14. From this point onward, any newly synthesized DNA would be light. Now, they simply allowed the bacteria to divide. To separate DNA by weight, they used density gradient centrifugation. Heavier DNA moved lower in the tube, while lighter DNA stayed higher. After one round of DNA replication, they examined the DNA from the first generation. Instead of seeing one heavy band and one light band, they found a single intermediate band. This immediately ruled out the conservative model, because if DNA had replicated conservatively, the original DNA would have remained completely heavy, while the newly made DNA would have been completely light. The centrifuge would therefore have produced two separate bands, not one intermediate band. But both the semiconservative and dispersive models still match the results. They let the bacteria divide one more time and examined the second generation. They observed one intermediate band and one completely light band. If DNA had replicated by the dispersive model, every DNA molecule would still have contained a mixture of old and new DNA, so there would still have been only one intermediate band instead of two separate bands. This result could not be explained by the dispersive model. It matched only one model, semiconservative replication. The experiment proved that during DNA replication, each daughter DNA molecule contains one original parental strand and one newly synthesized strand. This elegant experiment is still considered one of the most beautiful experiments in biology.